All right, let's get into it. Today, we are walking right on the razor's edge between extending life and the age-old dream of actual immortality. We're going to be talking all about cryopreservation, the real science, the wild speculation, and the very human story behind it all. I mean, this is the big one, isn't it? Could we really, one day, just cheat death by freezing ourselves? For decades, this has felt like pure fantasy, something straight out of a movie. But what's really fascinating is that it's moved from science fiction into a real and, a really controversial industry. People are actually paying hundreds of thousands of dollars for this tiny chance to wake up in the future. So let's really unpack this. This quote from the West Australian just nails the whole idea, right? It's this incredible, almost unbelievable promise. You freeze someone right after they're legally declared dead, and then you just wait. You wait for future technology to cure whatever ailed them and bring them back. But what does the science of today have to say about any of this? To really get a grip on the huge challenges here, let's first look at what's actually possible right now. We're going to put that dream of reviving a whole person on the shelf for just a minute and look at the proven science of preserving a single, incredibly complex organ. So for a long time, scientists have kind of worked under this rule that once a heart is removed, its cells start to break down almost immediately. And that makes things like sharing samples between labs really tough, and it means more animals are needed for research. But this one study asked a really simple but powerful question. What if we could preserve a mouse heart in cold storage for 24 hours without any major damage? The experiment itself was actually pretty straightforward. Researchers took some mouse hearts, put them in a special cold solution for a full 24 hours, and then they compared the cells from those hearts to a control group of fresh cells that were checked out right away. The whole point was to see if the cold-stored cells were still alive, structurally sound, and, functional. And check this out. This visual shows the results perfectly. What you're looking at here are microscopic images of heart cells. The top one, the control, that's a fresh cell. The ones below it, those are from hearts stored for 24 hours. And you can see that glowing green network. That's the T-tubule system. It's absolutely crucial for making the heart beat. It looks almost identical. The structure was totally preserved. Okay, so they looked the same, but did they still work? And what's so cool about this slide is that the answer is a resounding yes. These lines you see are action potentials. Think of them like the little electrical sparks that tell the heart to beat. The blue line is the fresh cell, the orange and red are the 24-hour-old cells. I mean, you can barely tell them apart. They didn't just look the same, they acted the same. So here's the bottom line. Scientists proved for the first time that you can successfully keep the structure and the function of a complex mammal organ for a full 24 hours. Now, for medical research, this is a huge deal. It could mean using fewer lab animals and making it easier for labs to work together. But, and this is a big but, preserving a heart for one day is a whole different universe from preserving a human being for centuries. So now, let's jump from that proven science to the much, much bigger ambition of cryonics. We're not talking about preserving an organ for research anymore. This is about preserving a whole person for revival. The key piece of tech that supporters say makes this possible is a process called vitrification. It's really clever. Instead of just freezing a body, which would cause ice crystals to literally shred every single cell, they replace the water in the body with a cryoprotectant solution. It's basically a medical-grade antifreeze. And this turns the body into a kind of glass-like solid state at super low temperatures, which in theory preserves its structure. And in the real world, this is what it looks like. After a person is declared legally dead, their body is filled with these special cryoprotectants. Then, it's slowly cooled down and placed, usually head first, into one of these giant steel tanks called a diwar. It's filled with liquid nitrogen, keeping the body at a mind-bogglingly cold -190 degrees Celsius. The hope, the big dream, as explained here by Peter Tolakidis from Southern Cryonics, is that future science will be so advanced that it can not only reverse this whole vitrification thing, but also cure whatever killed the person in the first place and fix any damage that happened during the procedure. It's really just a massive bet on the future of technology. But this is exactly where we run into this enormous gap between proven science and, speculative hope. The leap from that 24-hour heart to a 200-year human is, it's vast. This chart really lays it out. The 24-hour heart study, that was about short-term preservation for research, and we know it's possible. Human cryonics, on the other hand, is aiming for indefinite revival, which is, at this point, completely speculative. And the biggest challenge isn't just stopping decay, it's about reversing legal death, and maybe most importantly, repairing the unbelievable damage that happens to the brain. And that brings us right to the mainstream scientific view, put really clearly here by pathologist Dr. Marcus Dabner. See, the problem is that a person is already dead. Their brain has already started to degrade before they even start the preservation. A lot of scientists believe that reversing that damage, especially to the incredibly complex web of memories and personality in our brains, is fundamentally impossible. And beyond all the scientific debate, just the attempt to do this raises some huge financial, ethical, and legal questions for us right here, right now. First up, let's just talk about the price tag. In Australia, whole-body cryopreservation costs about $200,000. And that's not just a number, right? That's a decision that can completely change a family's financial future and what happens with their inheritance. And yeah, it often leads to some serious conflict. A really famous example of this is the case of baseball superstar Ted Williams. After he died, his kids got into this really bitter legal fight. One of his daughters wanted to follow his will, which said he wanted to be cremated. But his other two kids came forward with a separate note saying he actually wanted to be cryopreserved. It just goes to show you how these incredibly personal choices can tear families apart. And honestly, our legal system is just totally unprepared for any of this. Under common law, a corpse can't legally own property. So what on earth happens to a person's estate? And think about this. If someone was actually revived, they'd essentially be a cryonics refugee. No money, no property, totally disconnected from society. And to top it all off, the whole industry is barely regulated, kind of operating in a legal Wild West. So it all comes down to this one fundamental question. On one side, we've got proven science showing we can preserve an organ for a day. And on the other, we have a speculative industry selling the dream of revival in a few hundred years. Is cryonics the ultimate expression of our hope and technology? Or is it a fantasy that's just preying on our deepest, most human fear of death? That's the line we're all still trying to find.